Hello, everyone. Today, we are going to take a look at organic compounds. In our last discussion, we took a look at the chemical basis of life and understood the foundation of these organic molecules that we're going to talk about today being chemical elements. So let's go forward. So life's molecular diversity is based on the properties of carbon, right? So carbon's ability to bond with or other atoms is the basis for building large and diverse organic compounds. Carbon chains form the backbone of most organic molecules, and these molecules can exist as isomers, which have the same molecular formula but different structures. And then we also have hydrocarbons, which are composed of only carbon and hydrogen atoms. And so what we'll see here is that there are, from our last discussion, we identified the chemical elements that are most abundant in living things. And so that's going to be the basis of these organic compounds: carbon, hydrogen, oxygen, and sometimes nitrogen, right? So a few chemical groups are key to the functioning of biological molecules. And organic compounds properties depend on the size and shape of its carbon backbone and the atoms attached to its skeleton. So we have hydrophilic functional groups, which give organic molecules specific chemical properties. And so on the next slide, we have a table that's going to illustrate 6 important chemical groups that help to form these compounds, these organic compounds. So we have hydroxyl groups, carbonyl groups, carboxyl groups, amino groups, phosphate group, and methyl groups. And so as you continue to move through this course, you will see compounds with these structures pertaining. And so you should be able to identify the chemical groups that do not contain carbon in this case. So we see that in our phosphate groups, the amino groups, and so forth. The hydroxyl groups don't contain carbon. And so again, these are just a few of the chemical groups that we should be familiar with as it relates to these organic compounds that we're going to take a look at, these macromolecules. And so as I mentioned before, a few chemical groups are key to the functioning of biological molecules. So for example, the sex hormones, testosterone and estradiol, which is a type of estrogen, they differ only in the groups of the atoms that are highlighted. And so we'll see throughout this lesson how the structures of these compounds can change what they are. So in this case, we see testosterone and we see estradiol. Okay, the only difference here are the is this hydroxyl group here in the estradiol. Okay, so cells make these large molecules that we refer to as macromolecules from these smaller molecules or monomers. All right, so today we're going to focus on 4 classes of biological molecules that contain these very large molecules. All right, these are called our macromolecules, or sometimes they're referred to as biological molecules, but the term macromolecules refers to the fact that they are very large in size. And so we refer to them as polymers because they are made from identical or smaller building blocks that are strung together. All right, and so the building blocks of polymers are called monomers. All right, so you should be familiar with this relationship between a monomer and a polymer. All right, so for each of the 4 classes of biological molecules that we'll talk about, I want you to be able to identify the monomer, right? The monomers that are used to make these larger, more complex polymers. All right, so cells make these larger complex molecules from a limited set of small molecules. All right, so these monomers can be linked together through different types of reactions. We can have a dehydration reaction or we can have hydrolysis. And so these are going to either involve the use of water or the creation of water during the reaction. Okay, we know that these reactions are mediated by the presence of enzymes, which are our biological catalyst, right? So a checkpoint question here that you can think about is that suppose you eat some cheese. What reactions must occur for the protein of the cheese to be broken down into its amino acid monomers, and then for these monomers to be converted to proteins in your body. What type of reaction happens here? All right, this is just a diagram that shows us the dehydration reactions that can occur, and as well as hydrolytic reactions where water is introduced. Okay. So our classes of biological molecules. All right, let's start with carbohydrates. We are all familiar with carbohydrates as we mostly consume them daily. Carbohydrates range from smaller sugar molecules known as monomers to larger polysaccharides. All right, the sugar monomers that make up these larger polysaccharides are going to be our monosaccharide. A monosaccharide has a formula that is a multiple of the carbon, hydrogen, and oxygens, and contain a hydroxyl group and a carbonyl group. All right, so we want you to know that the monomer, the building block for large complex polysaccharides, are going to be our monosaccharides. Excuse me. Here we have honey as an example of a carbohydrate. All right. There are different examples of monomeric sugars here. We have glucose and fructose. So different combinations of these can yield other disaccharides and polysaccharides. All right. So we have some structural formulas here. I'm not going to necessarily hold you responsible for memorizing all of the functions or structures of these sugars, but you should be able to readily identify sort of a simplified structure of a sugar like this one, glucose, C6H12O6. Two monosaccharides are linked together to form a disaccharide. Okay, so two monomers can bond, right, to form a disaccharide in a dehydration reaction, and we see that water is utilized here. The bonding of two glucose molecules can yield the disaccharide maltose. Okay, so the nice question that comes up as we're talking about these carbohydrates and the consumption of carbs in our diet is, are we eating too much sugar? Okay, the FDA recommends that only 10% of daily calories come from added sugar. All right, research supports the correlation between high sugar intake and adverse health effects. Okay, checkpoint question for you to think about. Sugars are often described as empty calories. What do you think that means from a nutrition standpoint? What are empty calories? Here is a diagram that illustrates the amount of sugar that is annually consumed. So the World Health Organization recommends from a yearly consumption standpoint, shown with 5-pound bags of sugar, recommends about 20 pounds of sugar. The FDA recommends no more than about 40 pounds of sugar for an average yearly consumption. However, the actual yearly consumption of the average American is over 130 pounds of sugar. This is very alarming, and it has very significant implications for the health of Americans. So polysaccharides are long chains of sugar units. All right. There are other storage forms of these polysaccharides that we might find in plants and animals, such as starch and glycogen. Okay, cellulose is a structural polysaccharide found in plant cell walls, while chitin is a component of insect exoskeletons and fungal cell walls. All right, so you should be able to sort of compare and contrast starch and cellulose to plant polysaccharides. All right. So we have starch, glycogen, cellulose, all as examples of our complex polysaccharides. All right, let's look at our next biological molecule, next class of macromolecules, the lipids. Lipids are our fats, okay? Fats are lipids that are mostly energy storage molecules, all right? So in the absence of glucose as a preferred energy source, our bodies can utilize fats, okay? Lipids are diverse hydrophobic compounds composed largely of carbon and hydrogen, all right? So we're looking here and we see that these triglycerides or these fats consist of glycerol linked to 3 fatty acids, all right? So these are our building blocks of lipids, these triglycerides and fatty acids, all right? Some fatty acids contain 1 or more double bonds, and we'll be able to see some structures here shortly forming these unsaturated fatty acids, all right? Unsaturated fatty acids are typical of plant oils, all right? Fats with the maximum number of hydrogens are called saturated fatty acids, all right? Saturated fatty acids are found in the animal fats, okay? So we want you to be mindful of these 2 different types of fats and what their sources are, right? Hydrogenated vegetable oils are unsaturated fats that have been converted to saturated fats by adding a hydrogen. This hydrogenation creates trans fat, which are associated with health risk. So as you are consuming various foods and you're learning to read nutrition labels, you're looking to see the types of fats that these foods contain, okay? We want our unsaturated fats, all right? We don't want a lot of trans fats. So here's the structure of the glycerol with the fatty acid attached. Okay, these are our building blocks. These glycerol triglycerides are our building blocks for lipids. Okay, so we have our saturated and unsaturated fats. Again, we're consuming these in our diet daily, and so again, we want to be mindful of the types of fats that we are consuming and try to increase our intake of unsaturated fats as much as possible if we have to. And our bodies do need fats, okay? So, animation here for creation of these fats. Okay. So, scientific studies have documented the health risk of trans fats. Okay, so one question that you think about is what is the difference between a retrospective and a prospective study? All right. This image right here just kind of shows us the percent risk for health effects in terms of the types of fat that we consume. Okay, so the risk of heart disease and other factors significantly increase as you increase the amount of trans fat that you consume. At the baseline level, saturated fats are going to kind of be at normal or no risk difference, where we have these polyunsaturated and monounsaturated fats are going to help to reduce your risk of heart disease and other health complications. All right, so we want to try and consume these polyunsaturated and monosaturated fats. All right, so the lipids. All right, there's a special type of lipid that we want to be familiar with as it relates to biological science, and that is the phospholipid. Okay, phospholipids are major components of cell membranes. All right, there are also other lipids such as steroids and cholesterol. All right, steroids include the cholesterol and some hormones. Others are cholesterol is a common component in animal cell membranes and is also the precursor for making other steroids, including sex hormones that are necessary for sexual reproduction. So we want to be able to compare the structure of a phospholipid with that of a fat. Right, this phospholipid has a different structure where we've got these this what we refer to as a amphipathic molecule. This phospholipid has a hydrophobic head, okay, and this hydrophilic head and the hydrophobic tails. All right, and so when we create when the plasma membrane or the cell membrane is created, it is created with a phospholipid bilayer, where we've got sort of these two sheets of phospholipids creating this barrier around the cell, which is the cell membrane. And so we've got these hydrophilic heads that are interacting with the aqueous environment on the outside of the cell. And then we have these hydrophobic tails that are buried on the inside of this phospholipid bilayer that makes up the cell membrane. All right, we got our phosphate group here, we've got our glycerols, and we've got these tails. So if we were to look at the sort of a 3D structure of the cell membrane, we're going to see this phospholipid bilayer, where we've got these hydrophilic heads on the outside being exposed to water, and these hydrophobic tails are buried on the inside. All right, this individual structure right here is that of a phospholipid. All right, you should be able to readily identify a phospholipid structure and identify the components of this structure, where we have the hydrophilic head, which is polar, and the hydrophobic tails, which are nonpolar. Okay. Our next class of biological molecules are proteins. All right. Proteins have a wide range of functions and structures. All right. They are involved in nearly every dynamic function in your body and are very diverse. So we know that our proteins are ultimately the structural or functional units of our cells. All right. So proteins can have many, many different functions in our bodies. Right. They can function as enzymes. They can function in the transport of proteins embedded within cell membranes, so that cell transport. They can function as defensive proteins, such as antibodies, protecting our bodies from infections. They can function as signal proteins, such as hormones, participating in feedback and so forth. They can function as receptor proteins for communication, contractile proteins found within muscle cells, structural proteins such as collagen, and as well as storage proteins. And so we want you to be able to think about this broadly speaking, all right, the role of proteins in our bodies and how they can have a very diverse array of functions, all right. Proteins are composed of differing arrangements of a common set of 20 amino acid monomers. And so here we have our building blocks of proteins being the amino acid. Okay, there are 20 naturally occurring amino acids that occur in nature. And depending on the polypeptide sequence, the amino acid sequence will determine which type of protein is created. So the functions of different types of proteins depend on their individual shapes. And so in just a second, we'll look at the different shapes or conformations that proteins can take. And it is these shapes or arrangements of these proteins that will dictate their function. Okay. And so in the process of denaturation, a protein can unravel or change its conformation, loses its specific shape, and therefore the protein function can be altered. All right. So I want you to understand what happens during denaturation. If a protein is denatured, what happens? Okay. It's losing its original shape. All right. And we know that there is a direct relationship between structure and function of biological molecules. Okay. So checkpoint question: why does a denatured protein no longer function normally? Okay, we're thinking about here the relationship between structure and function. So protein diversity is based on different sequences of amino acids. So the monomers that are going to contain the amino group, a carboxyl group, okay, that hydrogen atom, that R group, these are all going to be attached to that central carbon and that are ultimately going to determine the reactivity or interactions of these amino acids. Okay, the R groups distinguishes the 20 amino acids, each with very specific properties, being hydrophobic, hydrophilic, and so forth. And so amino acid monomers are linked together in dehydration reactions, right? So we end up creating a peptide bond. Okay, so when we join the carboxyl group of one amino acid to the amino group of an adjoining amino acid, that peptide bond is formed, and this is how we get our polypeptide chains created. All right, so our monomers here again are the amino acids that are being held together by these peptide bonds in order to form our polypeptides, which will ultimately become our functioning proteins. Okay, so additional amino acids can be added by the same process to create a chain of amino acids that we call a polypeptide. So when you hear this term polypeptide, it is sometimes used interchangeably with a protein, but a protein indicates that it has its final functionality. And this polypeptide chain is just a sequence of amino acids in sort of a linear sense. So like the primary conformation here is a polypeptide chain. So question: by what process do you digest the proteins you eat into their individual amino acids? How do we process the foods that we eat? So this image right here shows you the basic structure of a protein. All right, we have an amino acid here, where we have the amino group, a carboxyl group, and we have this R group here, all attached to that central carbon. All right, there different properties of the amino acids are determined by its R group. All right, so we've got leucine here as the as an example of a hydrophobic amino acid, and over here we've got some examples of hydrophilic amino acids, serine and aspartic acid. Okay, so these are some examples of the 20 amino acids that exist in nature. All right, so we have these peptide bonds that are being formed here as amino acids linked together. Okay, so a protein's functional shape results from 4 levels of structure. There are 4 types of 4 levels of structure of the protein that you should be familiar with. Okay, so we've got primary structure, secondary structure, tertiary structure, and quaternary structure. Okay, a protein's primary structure is simply the sequence of amino acids in a polypeptide chain. So we've got this linear sequence of amino acids making up this primary structure. Okay, the secondary structure is formed due to the coiling or folding of that polypeptide chain, which is stabilized by hydrogen bonds. All right. Tertiary structure of the protein is the overall 3D shape of a polypeptide resulting from interactions amongst the R groups of different amino acids. And then finally, we have the quaternary structure, which is created due to the interactions of more than one polypeptide chain arranging into a quaternary structure. Okay, so a protein's functional shape. So we're saying that the function of this protein is ultimately determined by its shape or conformation at the primary, secondary, tertiary, or quaternary level. Okay, so a question: if a genetic mutation changes the primary structure of a protein, how might this destroy the protein's function? How does a mutation that changes the primary structure of a protein affect its overall function? So here, this diagram illustrates the primary structure, which is the linear amino acid sequence. The secondary structures we can create are examples are like the alpha helix and these beta pleated sheets. All right, and we have tertiary structures where we start to get this 3D arrangement of our amino acids. And then a quaternary structure here is created when you have more than one polypeptide chain in association, okay, into a functional protein product, right? So I would like you to be familiar with these 4 structural levels of the protein, okay? An animation here. All right, so our last class of biological molecules are the nucleic acids, okay? The nucleic acids, DNA and RNA, are information-rich polymers of nucleotides. So in this case, the monomers that make up the nucleic acids are nucleotides. Nucleotides are composed of a sugar, a phosphate group, and a nitrogenous base. I do want you to be able to detail the structure of a nucleotide, as the nucleotides are the building blocks of nucleic acids. DNA, one of the nucleic acids is a double helix, okay? This is going to be 2 strands of nucleotides that bind around each other in sort of a helical formation. RNA, on the other hand, is a single polynucleotide chain. And DNA and RNA then serve as the blueprints for proteins and thus control the life of the cell. So DNA is the molecule of inheritance. All right, so you probably heard the phrase that DNA is the blueprint for life. It contains the instructions for creating proteins, right? Which ultimately determine the function of our cells. And so we'll talk in another section or another chapter about the central dogma and how we use the information contained in DNA to transcribe RNA, and then ultimately that RNA will become a message and will be translated into proteins, right? And so DNA is the molecule of inheritance. This is the genetic information that's passed down from parent to offspring during reproduction, okay? So the structure of the nucleotide, we've got a sugar group, right? In DNA, it is deoxyribose, which we have this oxygen missing on this 2' carbon here. We got a sugar group, 5-carbon sugar, which is pentose. We've got a phosphate group here, and we've got a nitrogenous base. There are 4 possible nitrogenous bases that make up this DNA molecule. And this double helix that we mentioned before is held together due to hydrogen bonds, okay, amongst these nitrogenous bases. All right, so we refer to this structure as having a sugar phosphate backbone. All right, so there is adjoining sugar phosphates being held together by phosphodiester bonds in order to create this entire strand of nucleotides. All right, so we have our sugar group, phosphate group, and then our nitrogenous base here in this case, which is adenine. Okay, there are 4 nitrogenous bases in DNA that you should know: A, T, C, and G. Okay, the nitrogenous bases on one nucleotide strand will base pair with the nitrogenous base on another nucleotide strand in order to make this double-stranded DNA molecule. Okay, and so we see here this base pairing occurring. This base pairing occurs in a predetermined manner naturally. A complementary base pairs with T, and C complementary base pairs with G. This happens and it creates this double-stranded helix for our DNA molecule. Okay. And as I mentioned before, DNA is the blueprint. It contains information or instructions, and it is arranged into, we'll say, informational units that we call genes. All right. These genes will ultimately encode proteins, which therefore determine the function of our cells. And so the information contained in our DNA is quite critical. All right. So the process of transcription and translation is how we take the information in our DNA to create RNA, which is an intermediate molecule, which will ultimately be used as a message and decoded in order to produce protein. All right. So we have our 2 types of nucleic acids here, right? We have DNA and we have RNA. All right, you should know the difference between these 2 molecules: DNA as a double-stranded helix and RNA as a single-stranded molecule. Okay, you should know the nitrogenous bases of DNA, which are A, T, C, and G. And then in RNA, those nucleotides differ. There's no thymine in RNA. Okay, the T that we find in DNA is replaced by uracil in RNA. Okay. So as I said before, the nucleic acids, DNA and RNA, are information-rich polymers of nucleotides. Okay. So checkpoint question: what role does complementary base pairing play in the functioning of DNA? So we have checkpoint questions here that I want you to work on and submit by the end of the week. You should be able to describe the importance of carbon to life's molecular diversity. All right, we talked about carbon being the backbone of all organic molecules. All right, describe the chemical groups that are important to life. So I showed you a table where we listed some of the important chemical groups, like the carboxyl group and the carbonyl groups and so forth. All right, explain how a cell can make a variety of large molecules from small sets of molecules. All right, so we talked about the 4 classes of macromolecules and what their monomers were, or the building blocks for each of those classes. Okay, define monosaccharide, disaccharide, and polysaccharide and explain their functions. All right, define a lipid, the phospholipid, steroids, and explain their functions. All right. You should be able to describe the chemical structure of proteins and the importance of proteins to cells. That is certainly an important question here. And lastly, you should be able to describe the chemical structure of nucleic acids and explain how they relate to inheritance. And we will stop here for today.